Dr. Ramesh Sistla is a distinguished expert in computational drug discovery, combining a rich academic background and industry experience to drive innovation in molecular modeling. He is the founder and managing director of Think Molecular Technologies Private Limited. With a PhD in condensed matter physics from IIT Madras and postdoctoral research in computational biology at IISc Bangalore, Dr. Sistla brings a deep understanding of molecular interactions. Now I will introduce Dr. Veeran. Dr. Veeran is working as a research investigator in Think Molecular Technologies Private Limited for the past 4 years. He has a PhD in computational physical chemistry in the field of thermodynamics and kinetics at the Indian Institute of Technology, Chennai. Let us all welcome them to the stage with a round of applause.
Hey, good morning, everyone. It's nice to see it's more people than 2024. So welcome everyone to this session, to my talk especially. Glad to be here. I think Team Advent has put up a pretty good show, as we can tell from the number of calls we have received before the meeting, which means that now they are, proactively organizing this and reaching out. And 26 academic institutes they have reached out. I think Surojit is traveling quite a bit, it looks like. So good. So we hope to move to a bigger venue next time, of course in Bangalore. Okay. Okay. So nice to be here. Over the next, so we are doing I am opening the innings now. I am taking strike, but soon Velan will take over. Okay. So I am here to do a little bit of introduction on what we do. And then, of course, this is an ICM user meeting. So Velan will take it over and then tell you how our team has put ICM to use in trying to design diverse modalities. Okay, we have not done biologics, by the way, but it's all small molecules. Okay, so Think Molecular is what it is. I'll tell you in a bit. We do anything that has to do with small molecule drug discovery. Okay, in small molecule drug discovery, starting with target identification all the way until we do full scale drug discovery partnerships in the design space. That means before the experiment, we do all the theory and computation and keep informed our partners. No matter what it is, we actually can weigh in computationally for that. Okay, so all the way from target identification to lead generation, lead optimization. And it's not just that, we want to solve exciting life science problems. Okay, so we also make use of the various technologies that we have in the space of chemical biology. We have done projects where our partners had assets that they were in licensing. So we do the technological due diligence for that. We identify the data gaps and finally give a recommendation of whether it's a good thing to in-license or not. Or if you want to out-license something, we are there to tell you what can bring you great value for that. We do target dossiers for new drug targets, basically telling people that if you want to start a new drug discovery project, what it takes, okay, what are the different experiments to do, what are the kill experiments to do in a given target. So you just tell us the therapeutic area, we can do that. So because our team has both computational chemists, AML, biologists, everybody. IP generation is at the heart of what we do, and we, of course, doing library design all the time. And everything is powered by our AML augmented, physics-based simulations. Next, please. And we are actually an NVIDIA Inception grantee. So which means that we are in the NVIDIA ecosystem trying to leverage the strengths of hardware and software and of course the VCs that NVIDIA actually provides. So we compute with in-house computing power as well as on the cloud. And in terms of the software, we use a collection of proprietary tools like Molsoft that we license tools. We use our own in-house tools which are proprietary to us, and also a fair bit of open source tools. And we have set up over the last 5 years sufficient drug design expertise, and we are able to scale at will. Okay, so in terms of so we are an active partner for a lot of biotech companies. Like you can see, we have a global spread. We are not in Africa yet, but we'll get there soon, I hope. So we do a fair bit in India, and also in the east and west coast of US. We have multiple collaborations going on. In fact, in the last 5 years, we had more than 20 clients with whom we have actually worked. So, which is a kind of validation for me coming out of after kicking a good job elsewhere. Yeah. So, and yeah, so we have even got onto the patents that the partners have published. So, which is some validation that we have got something right. And our business models are FT or fixed price or even a bit of risk sharing. So more recently, we have actually got into this space where we also collaborate with synthesis partners or even biology partners. So we are able to do, we design, they synthesize, and then we can give the whole thing to the partner actually. So which has helped our partners to boot their targets, the new targets in a much faster time frame. So then next fellow. Yeah. So basically, drug discovery is a design, make, test cycle. So design is think molecular, right? So that's what we mean. That's what you see here. Yeah. So in short, if you have a therapeutic hypothesis, we have the computational fluency to get you the chemical matter. That's too much of a sales pitch. So what do we actually do, right? So when I thought of think molecular, I actually didn't want it as a therapeutics company or something like that, but more as a technologies company. Okay, so we are a technologies company in the space of life sciences and drug discovery at the moment. Everything is done by molecules, so drug design won't be limiting for us. We can basically look at anything in the future. I don't want to read through the whole thing, but you can see that there are very familiar things like molecular docking or molecular dynamics and analysis. And there are also very new things like LLMs, machine learning, deep learning, knowledge graphs. So we basically practice all of these. Okay, there is not one thing here that we don't practice. So we practice all of them. So how are we relevant here? So all the boxes that we have here, that's where ICM provides these technologies. Okay, so we have been using one or more of those pretty routinely and in a fit for purpose manner. So we don't use a technology because we have it. Based on the question that is being asked to us, we actually practice a certain technology. And so this also tells you we do a fair bit out of ICM also. Okay, what ICM doesn't offer yet, we do those also. So and of course, Ruben's that particular slide was very thoughtful, where he showed only 12% that the deep learning models agreed with the the actual physics based models. But the thing with machine learning and deep learning is that the improvement curve can be pretty dramatic and rapid. So next year when you do it, it could well be 50%, right? So, okay, these are the various technologies that we use for drug discovery. And so what we have achieved, so we have been very engaged with science. So you can see in the number of publications that we have, from book chapters to, invited reviews to other peer-reviewed publications. And more importantly, we have played a key role in the NCE discoveries of at least 3 to 4 different partners. And last year, one of our partners presented a first-in-class PARG inhibitor in the ASER conference. And of course, we have got co-inventorship on the patent. So this is a work that is actually done by our entire team. Now, Velan will give you glimpses into, the different things that we have actually done. It is not meant as a very deep dive into anything, but as a how you can, because I have been the user of a very popular and very expensive tool for a long, long time, like maybe some of you also were, okay. So now after I actually took to ICM, I was pretty enamored by, the diversity of what it can actually accomplish. I am a big believer in molecular recognition, and I think with ICM, I see a lot of possibilities into molecular recognition. So we will give you some glimpses. Velan will talk about a few of the case studies that were done by our whole team over the years. Not comprehensive, but just we kind of chose to give you a feel of what it is. Well, take it away. I'll come back to answer any of the questions if you have at the end of it. Thank you.
A very good morning to all. Today I am going to talk briefly about our efforts in various diverse drug discovery efforts in diverse modalities like small molecule discovery, covalent inhibition, and structural models involving the metals, and drug discovery efforts in peptides and protein models and RNA binders. And how we used various ICM tools in designing. In any initial part of the drug discovery program, we looked into the literature to gather information for any particular target proteins. We identify the interaction fingerprints from the reported PDB database and understand the comprehensive structure activity relationship information from the reported structures. And we generate atomic property field based pharmacophores from the reported ligands incorporating various chemical series of any particular target proteins. And we screen against large databases which we have access over to 300 million compounds with pre-generated conformers. We screened against those large library virtual databases and obtain virtual hits for the new chemical matters. We also employ fragment-based drug discovery efforts to identify new chemical matters, where we probe the binding site of any target protein using fragments. At Think Molecular, we have some 2000 plus fragments which were derived from reported lead compounds, which has diverse fragments like hydrophobic, aromatic, heteroaromatic like fragments. So here we have put it representative figures, where we probe the binding pockets through various fragments to understand or identify the interaction hot spots in a particular binding site, and hybridize these fragments using suitable linkers to obtain new chemical matters. So we also have close to 4 million plus virtual ring systems which were developed by Peter et al. Using these methods, we obtain new hits chemical matters for any target protein, and we take it into the structural models. For any target protein, any different diverse chemical compounds can be modeled for any particular protein. Here, for representation, the binding site residues are shown in the yellow color with one particular conformation of each residues. So this particular conformation can give us structural models for any one specific type of chemical series. So it may not be ideal for some other different chemical series of any target proteins. So we leverage the diverse conformations reported in the PDB database for any target protein. So we incorporate those other conformations also for our structural models, which to make an ensemble grid for the structural models, which are also known as the 4D grid. So this is to incorporate the flexibility of the binding site residues into our structural models and to improve the accuracy of our models. So this is a video representation to show that how we incorporate various conformation of the binding site residues in our structural models. We can also incorporate the flexibility of any one important residues in the binding site, which are considered to be particular target protein. So we can also flexibility take into account one flexible residue also. This is one small case study where we show the advantage of using the ensemble grid or 4D grid. The first picture where we model a set of reported ligands for in one rigid grid with only one conformation of the binding site residues, where the distribution of scores, it is minus 30 to 0, the docking score is. Suppose once we model in the 4D grid with diverse conformation of the binding site residues, as we can see, it is the docking scores improve to less than 40, minus 40 also. So it is a clear improvement in the docking scores, and it will improve the docking models of all structural ligands. We also use a tool named 3D ligand editing, which from the ICM, which we widely use in all of our drug discovery programs. The advantage from this method is, for example, in our lead optimization process from our initial chemical matter, if you want to optimize a particular group, for example, here I have boxed one particular group. So the advantage of this 3D ligand editing is that we can replace or optimize a particular group in presence of the protein. So in this particular example, if suppose with respect to protein, we are replacing one group from the new chemical matters. From this, and generate new libraries using our in-house fragments as well as ICM fragments or vendor fragments. We can generate large libraries for replacement of any chemical groups. So this is the pictorial animation where we can generate new libraries. This is with respect to the presence of the protein. Once we identify a new compounds from that, so we rigorously model or more refined with further structural models also. Another tool we utilize in some of our programs is an exit vector prediction from using ICM. So for any optimized ligand, if we want to grow a particular ligand to the further grow to the solvent leading to the solvent space, or this particular tool will help us to identify the hydrogen atoms which are suitable for the substitutions. So in this table, this tool particularly scans the hydrogen atoms of any chemical species and lists the rank accordingly based on their access to solvent regions. So here the top scoring hydrogen atom, which is H222, it is shown in the green color bulge in the figure. So that is the hydrogen atom which is most favored to substitute and grow the molecule into the solvent region. So we utilized this tool in a couple of our programs to design chemical probes for novel assays and to design radiopharmaceutical ligands. And it has many more applications also. So in one of our earlier projects, the target protein has no experimental structure. So we utilized homology modeling to predict the structure and modeled our earlier ligands through this homology predicted structures. Then these predicted structural models were later validated by the experimental structure. It was resolved. So we scanned that chemical ligand for the exit vectors to grow and design the chemical to attach the chemical probe. The phenyl ring is identified to attach the best position to attach the chemical probe. So we designed a fluorophore based probe, which was utilized widely in the later stages of that program. So this is the structure of the designed chemical probe. In another program which we utilize that exit vector prediction is the drug discovery program in identifying a radioligands to targeted in the targeted cancer therapy. So the radioligands ideally it has have to discover a ligand for a any target protein, a potent ligand will be designed, and it will again we will scan for the suitable hydrogen atoms to attach or grow the linkers and to attach the payload. Payload is not payload is the chemical entity which has a chelator molecule which coordinated to a radionuclide. So this particular radionuclide will bind to the target protein and emit radiations to destroy the cancer cells. So this is ICM enabled us. Here we have shown one particular example of a chelator which is known as TCMC. So ICM enabled us to draw this structure and model to get structural models with full radioligand. This is a structural model we obtained for one particular target protein where we identified discovered the potent ligand and grow with attached with suitable linker and then attached to a suitable payloads. The radioligand we help discover will be in the clinic soon. And the limitation of this structural in the ICM is we obtained the structural models with unrealistic docking scores as it involves a metal atom which may not be still parameterized, very well parameterized in the models. We worked on another project to target protein which we designed with the covalent warheads. So covalent inhibitors generally expand the target druggability space, and we discovered the first-in-class PAR covalent inhibitor targeting the cysteine, which was presented last year at the AACR meeting. So we also, in a couple of programs, we targeted proteins with trapping tyrosine and lysine through suitable warheads. So this is the pictorial representation where we proposed the covalent trapping of tyrosine as well as lysine through SUFX warhead, sulfonyl fluorides. ICM offers us traditional well-characterized warheads as well as any new warheads, newly developed warheads. We can define the reaction mechanism to model those new warheads also. We also worked on a target protein where it contained a catalytic metal atom. For example, the target protein has a catalytic zinc. So the ligand should contain a suitable metal binding group to get ideal potency. So in this particular case, the pKa prediction of the metal chelating group is significant. So here I have shown an example docking mode which we initially obtained from ICM, which is the zinc. The target has a catalytic zinc in the binding site. So we modeled through a sulfonamide group, which is a well-characterized zinc binding group. So our initial model showed that the sulfonamide oxygen is chelate to the zinc. However, in the sulfonamide group, the ideal zinc binding atom is the deprotonated nitrogen atom will chelate with the zinc. That is the ideal mode. So this particular docking mode was not ideal. So we forced in the ICM auto prediction of the pKa, the sulfonamide molecules, it does not predict the pKa of the compound very efficiently. So we forced that our initial designs, sulfonamide containing ligands with we forced it to predict put the NH minus. We fixed that and it modeled through the ICM to get the ideal mode, which where NH minus is coordinates to the zinc metal. So hence ICM pKa model needs updation, and we also put one example compound where fluorinated sulfonamide groups, ICM predicts it to be a 9.7. However, when we use another prediction tool, it is closer to 7. Its pKa is closer to 7. So this is one limitation we observe. This is a pictorial representation where we help discover a small molecule for a zinc containing target protein. We also worked on a target protein where its binding site is very shallow and hence we cannot target through ideal small molecules. So we targeted that particular protein using peptides. So from the initial hits from the experiment, our partners were able to resolve the crystal structures and characterize the peptide backbone, which we have shown in the magenta color with transparent. So once we characterize the peptide backbones of the peptides, so in our further refinement and improve the optimization stage, so we employed a template docking method in the ICM where we don't want to sample the peptide backbone. So we constrained the peptide backbone to the crystal structure backbone conformation, and we only sample the side chain amino acids to improve the potency. So without template docking method, we were mostly obtained with poor modes. With template docking, we were get more accurate models out of ICM. So we helped discover a potent macrocyclic peptide using the docking models as well as MD simulations and non-natural amino acids and peptidomimetics. We also worked on PROTAC modeling using ICM PROTAC script. So we validated our model initially with the experimentally reported structure for a bromodomain 4 degrader using a VHL ligase. So we also validated the structural models of 30 plus PROTAC mediated complexes reported in the PDB database. So the ICM ranked the different poses, various poses of this PROTAC complex. So we did not always observe the experimental mode in the top ranked mode poses. Sometimes the low ranked modes are also lead to the experimental results model. So we also predicted the docking mode structural models for clinically reported PROTACs. So here some 4 structural models we have listed. The first structure is the model for a KT474 PROTAC for IRAK4, which has implications in autoimmune and inflammatory diseases. The second top right one is the KRAS G12D mutation PROTAC, which has implications in the cancers. ARV110 is the PROTAC for the androgen receptor, which has implication in the prostate cancer. And ARV471, which has identified for the estrogen receptor, which has implication in the breast cancer. So it has granted fast track designation by the FDA. So as and when the experimental crystal structures are reported, we can validate these predicted structural models. We also worked on RNA splice modulators, which is a hot area of research currently. So the left-hand side animation shows the structural model generated by us for risdiplam, which is a known splice modulator along with the nucleus. So directed small molecules which can bind to the RNA at the splicing junctions and influence the protein expression of any target RNA. So we helped discover a compound that stabilizes an adenine bulge of an RNA, and it went through advanced profiling and it showed efficacy in cancer. In another program, we target the local binding pockets generated formed due to the secondary structure formation of the RNAs. These secondary structures can be targeted through small molecules and influence the expression of that particular protein. So we have designed potent compounds to bind in this particular pocket, which have been validated by SPR assays. So we wanted to build a database of compounds to target to bind with the RNAs. So we made use of literature where it lists the chemical properties of any compound and ideal properties for compounds to bind to the RNAs. So some of the properties listed, I have listed here. So the fraction of sp3 hybridized carbon, number of oxygen or nitrogen atoms, and basic pKa, and number of aromatic rings, and as well as the number of hydrogen bond donors in a particular compound. So we developed a scoring, MPO-based scoring scheme to formulate and identify compounds which can potentially bind RNA. So this table implicates an example range of the properties which can ideally bind with RNA. So we have developed many scoring schemes. Here we have shown only one sample scoring scheme for our. So this picture shows for an MPO scores for an example compound. So then we move to generate the MPO scores for a database of compounds. This is the distribution of MPO scores for full database. So ideally the compounds scoring MPO scores greater than 8 and 9 should have a potential RNA binding applications. So this is our group. I thank Dr. Ramesh for this opportunity to present here. And I also like to thank my colleagues for their help and support to put together this slide, as some of their work I presented were done by them. And also I will thank you all for your kind attention.